Alright, so let's take a look at mRNA processing now. So we've talked about transcription in prokaryotes, we've talked about transcription in eukaryotes, and we said in eukaryotes that the mRNA that we first produce is called a pre-mRNA. It's a baby mRNA, and it has to be processed before becoming an adult. In prokaryotes, we produce an adult right off the bat. Alright, so no processing involved in prokaryotes. And we do this in order to allow ourselves an additional level of control, right? So remember I think I told you guys this analogy. I love this analogy, by the way. Prokaryotes they don't really have to carefully control their gene expression very much. They're kind of like hermits living out in the woods, right? They can make noise, they can shoot guns into the woods, they can shoot off firecrackers, right? And nobody really cares. Single-celled, right? Whatever they do is only for them. Whereas with eukaryotes, we tend to be multicellular, so our cells are kind of like living in a high-rise with very, very thin walls. There's 10 trillion of our cells packed together. So we have to very carefully control our gene expression, and this is one way that we control our gene expression is through mRNA processing. Another way was actually through using those specific transcription factors that we just talked about. So let's actually take a look at mRNA processing now. There's 3 steps. We have to add a 5' cap. We have to splice out the introns. And we have to add a 3' poly A tail. Alright, so let me draw a little diagram for us here. Actually, let me get a little bit more complicated. Here's my template strand of DNA. In green is my promoter. And I'm going to draw in dark blue and then in light blue. You can probably guess what I'm meaning this to represent. The light blue are introns, and I'm just going to label them I from here on out. I. The dark blue are exons, not to be confused with the oil and gas company. Alright, and it would help if I could stay on the slide, wouldn't it? Okay, so when we transcribe a piece of mRNA, we go all the way from transcription start right there, we call that +1, all the way to the end of that gene, usually about 150,000 nucleotides. We include both the introns and the exons. We include both the good stuff and the junk. Remember we said that we thought introns were junk? At least that's one of the current hypotheses. You know, actually, it's going to take forever, isn't it? Let me do this. There we go. You can barely see the dark red, but you get the idea. So exon, 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 exon. Oop. Intron, 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 intron. Okay, so this is our pre-mRNA that we've made. Remember, not yet an adult. What we will do to this pre-mRNA is we will add on the 5' end of that pre-mRNA, let's say that for the sake of argument that this left end that I'm hovering over here is the 5' end. We're going to add a cap. And the cap is not like literally a hat. It's actually a modified G nucleotide. So we take a ribonucleotide, a G ribonucleotide, we chemically rearrange it a little bit and we stick it on the 5' end. On the 3' end, and again for the sake of argument, let's say it's over here, we add on what's called a poly A tail. That just means 100 to 200 A's. Right on the end there. And then as our last step, we will remove the introns, delete them out, and glue together the exons. So again, let me actually number those exon 1, 2, 3, 4, and 5. For the sake of completeness, 1, 2, 3, 4, and 5. Dang it. There we go. So now we have exon 1, 2, 3, 4, and 5. I keep doing that, don't I? Right there, along with our 5' cap and our 3' poly A tail. Okay, so this is our fully adult mRNA at this point. I'm about to tear up my hair over that. This is our fully adult mRNA at this point. Alright, we fully processed it. The removal of the introns is called splicing. Splicing. So we're splicing out the introns. Alright, and we'll do that because, again, we think that the introns don't really serve a purpose. At least, not one that we can readily see. The 5' cap, though, does serve a purpose. It helps ribosomes attach to the beginning of the mRNA to begin translation. The 3' poly A tail also serves a purpose. It slows the degradation of the mRNA. Here's the thing about mRNA inside our cells. mRNA inside our cells does not last very long. That's because we have a special set of enzymes called RNases that chew up mRNA. You can kind of think of them as little Pac-Men. It's a dumb-looking Pac-Man, but there you go. And they'll chew up RNA starting at the 3' end. They don't really chew it from the 5' end. We won't get into why. And we produce these RNases as a sort of, well, it is as a defense mechanism. First of all, a lot of viruses that infect us are made in part of RNA, so it's kind of a, the best defense is an offense strategy sort of deal going on here. Right? So we're preemptively destroying any viruses that happen to get in. And it also prevents RNA from hanging around inside the cell for too long and getting transcribed or translated too much. You see that? I made that mistake. It prevents mRNA from being translated too much. And it prevents us from making too much protein. So it's an additional regulatory step, these RNases. So here's what that looks like. Again, here's our DNA, exons and introns, all in there. We transcribe, we cap it, we tail it, and we splice it. And we end up with an adult, there we go, an adult mRNA, fully mature mRNA. And at this point, this mRNA will exit out of the nuclear pore and into the cytoplasm where it can go meet up with the love of its life, a ribosome. Oh, you thought we were done, didn't ya? No way, man. We gotta look at how splicing occurs. So first we're gonna look at the enzyme that does splicing and how it's made, and then we'll look at the process. So here's the process of mRNA splicing. So in order to make the enzyme that does the splicing, and by the way, the name of that enzyme is called a spliceosome. Love the name, right? Spliceosome. The spliceosome is made of a couple different things. It's made of a combination of RNA and proteins. Sounds familiar, doesn't it? Well, it's kind of made of the same stuff as a ribosome. RNA and proteins. Now, whereas a ribosome was made of rRNA, a spliceosome is made of something called small nuclear RNA. The proteins have no special name, though. Together, the combination of snRNA, small nuclear RNA, and proteins is called a small nuclear ribonucleoprotein, or snRNPs. Now, the short name for snRNPs are called SNRPs. You heard me right, they're called SNRPs. I love that name. SNRPs. SNRPs. Okay, I'm done. So SNRPs make up the spliceosome. and the spliceosome is actually what does the splicing reaction. So we actually see an image of a splicing reaction down here. I'm going to come back to that because that image will make more sense once we look at the mechanism. Okay? So again, we're taking a bit of DNA, transcribing it into pre-mRNA, and then we're splicing out the introns to get the mature mRNA. We've left off our cap and our tail in this particular image for dumb reasons, I'm sure. Okay, so over here we see one intron. So we're just going to focus on one intron here. I think we splice all of our introns pretty much simultaneously, although I'm not exactly sure. Our SNRPs are a combination of RNA and protein, assemble themselves at the intron-exon border, and usually we have a G and a U there. Right, so usually we have a G and a U, not always, but usually. And then somewhere downstream on the intron we will have an A. So this G, U, and A are all highly conserved within between introns. Now these SNRPs come together to form the spliceosome, and the spliceosome links the A with the U. Remember, A pairs with T, or A pairs with U in RNA. And the spliceosome will cut between the G and U and link the G to the beginning of the next exon. So again, this is exon, let's say exon 1, this is exon 2. And it forms a little loop right there. That little loop that's formed has a very technical name. It's called a lariat. You know, lariat, like the rope loops that cowboys used to use in those old movies, right? So the spliceosome will form a lariat, and it'll take that lariat out and do something unknown with that intron, that former intron. We're not quite sure what, leaving behind the joined together exons. Now, what's really interesting about this process is by the way, we are constantly discovering new and interesting and very exciting ways that spliceosome works. And in fact, some research suggests that the spliceosome may have actually evolved from an intron itself. It's made of RNA, right, and protein. And guess what? These introns right here are also made of RNA. How it works, though, exactly, we're still not quite sure. It is one of the most complex enzymes known, though. And I think one of the biggest. So when we look back here at this image, what we are seeing outlined in orange right there, that is all of the exons that are going to be joined together, looped out into these lariats. Those are the introns. Those are the introns right there. And I know it says single-stranded DNA, ignore that part. They used a certain, the people who took this image actually used a certain technique using DNA to demonstrate this. Alright, so here on the original graph we can actually see those introns looped out right there. Pretty darn cool, huh? In this image we do not see the spliceosome. It's been removed just so we can see the DNA and the RNA. Okay, so this is what our mRNA, our adult mRNA, looks like at the end, a closer look. Alright, so here's our, right here, there's our exons. Don't worry about this part where it says 5' and 3' untranslated region. We won't worry too much about that. Here's our 5' cap. Remember we said it's a modified G nucleotide, and it actually has 3 phosphate groups, so it's a modified GTP. Here's our poly A tail right there. This is actually going to be 100 to 300 A's long. There's our poly A signal. This is actually the remnant of our transcription stop. Remember with our transcription stop, it was a poly T in DNA, which would have become poly A in RNA. Alright, so if you're an RNA, you not only start with a bunch of A's as your transcription stop, during processing you add a couple hundred more. Now what's really cool about all this is that especially this 5' cap sort of acts like a ticket, right? It's like an entry ticket into the ribosome, the party that is the ribosome. And you need a 5' cap in order to be translated if you're an mRNA. You aren't going anywhere unless you get a 5' cap. Now what viruses do, assuming that they evade all of your defense mechanisms, is once they get inside your cell, they'll grab a nearby mRNA, they'll snag it, they'll chop off the cap and destroy this mRNA. They'll keep the cap and they'll add it to themselves. So certain viruses do this more often than others. Orthomyxoviruses, which is the virus that causes the flu, actually steals your 5' caps off your mRNAs when it gets inside your cell. I think I have a picture, yeah. So this is the enzyme right here, right here, right here, and right here from three different types of these viruses. This is the influenza virus, the one that causes the flu right there. All these others are not influenza virus, but they also cause illness. And this enzyme is what actually steals the cap off of your mRNAs and adds the cap to the virus's own RNA. So the cap, it essentially scalps your tickets, right? That's what influenza does. And this process is called cap snatching. So the flu virus snatches your 5' caps. That's the technical term, by the way, I promise. It's called cap snatching. Alright, and by the way, the family of viruses that the flu virus is from, it's called orthomyxoviruses, which is why I have that up there. Okay. Now here's one thing that is very, very interesting. That spliceosome, that big complex spliceosome, doesn't always follow orders. So sometimes, and let me, again, allow me to draw this out for you. Here's a pre-mRNA that I'm drawing here. Exons in dark red. Exon 1, 2, exon 3, exon 4, exon 5. And then introns in, actually let me do it in orange. A little bit obvious. I don't know about you, but I couldn't quite tell the difference in the previous slide. The difference between the dark red and the light red. So during splicing, we kind of go, okay, join exon 1, exon 2, exon 3, exon 4, and exon 5 together. Good to go, right? Not quite. So every once in a while, the spliceosome likes to get a bug up his rear end. And he will join together, say exon 1, then exon 3, then exon 2. Or he can make another version. He can make exon 1 and say just exon 5. Or he can do exon 4 and exon 2. Or exon 5, 4, 3, screw 2, just go straight to 1. Right? So this rearrangement of exons allows you to make a lot bigger variety of mRNAs, and this is called alternative splicing. Alternative splicing. So this means that one gene can produce lots of different mRNAs and therefore lots of different proteins. And this makes sense when we look at our cells, right? We have about 20,000 genes, plus or minus, and yet we have about 100 to 150,000 proteins, right? So this suddenly explains why we can make 100 to 150,000 proteins off of just 20,000 genes. About 90% of our genes will undergo alternative splicing. And this explains how we can make one version of a protein that just ends up in the cell membrane, and another version of that same protein that ends up secreted outside the cell. Right? You're just changing the protein trafficking tags through alternative splicing. You can make lots and lots of different antibodies capable of recognizing almost anything in the universe simply by reshuffling around some of your exons. Different exons may code for different subunits of a single protein, which allows for very, very, very fast evolution. Instead of reinventing the wheel every time you want to make a new protein, all you got to do is just shuffle around some parts that you know already work. All you got to do is shuffle around some exons. So this is what we see here. Okay, so here's our DNA. There's 5 exons. In between right here, this is introns. Here's our pre-mRNA right there. And through alternative splicing, we can produce one version that has all 5 exons in order, and we can make this protein right there, this shape. We can make another mRNA that's missing exon 3, and because it's missing exon 3, it's missing that part of the protein. Alright, so it'll have a slightly different shape. We can make yet another mRNA that's missing exon 4, so it'll be missing this part of the protein, and yet another slightly different shape. So again, the take-home message here is we are reworking something that we know already works. It's not very high risk. It allows us to adapt and evolve fairly quickly, faster than we would if we would just relied on pure chance. Okay, so one really cool example. Parasites like malaria. Malaria is a eukaryote. Oops, helps if I can spell eukaryote right. Malaria is a eukaryote. It's actually a protist. Malaria is one of the biggest killers on Earth. And one of the difficulties with malaria in treating it is that it evades your immune system. So one of the characteristics of malaria is that when you are infected, you go through these spikes of illness where it gets really bad and then you get better. And then it goes through another spike where it gets really bad and then you get better. And then it goes through another spike and then you get better. So it's this back and forth between your immune system and the malaria protist. Now, how the malaria protist does this is it uses alternative splicing in order to generate different proteins. So once our immune system learns one version of the malaria proteins, well, that malaria protist will just alternatively splice and produce a different version that our immune system no longer recognizes and can't attack. So that malaria is free to reproduce and rise and cause horrible symptoms, and then our immune system will learn to recognize it and start to attack it and destroy some of it. So then that malaria will lie dormant for a little bit, and then it'll alternatively splice again, it'll rise again, and then our immune system will learn it and attack, and it'll go dormant. So this is actually what in part explains that cycle of symptoms. So this is called antigenic variation. This process, an antigen is any molecule that triggers an immune response. So you're varying your antigens in order to evade the immune system. And this makes it really hard to develop a vaccine, because the whole point of a vaccine is to train our immune system to recognize something. And if that thing is always changing, well, it's hard to train. It's hard to train for. And I know that for us, this is not terribly that exciting, but here's the spliceosome of a malaria parasite, and here's the spliceosome of humans. We can see the RNA in black, and then the proteins in pink and orange and whatnot. Okay. So, again, you've gone from pre-mRNA to fully adult mRNA now. Alright, and that mRNA is ready to export. Looks like a little turd, because it's kind of folding on itself, and it has to get through the nuclear pore. So that's all this slide is showing, is export through the nuclear pore. Okay. So, let's see, what do we want to do here? Just some kind of little fun facts at the end. You don't really need to know these, but they're neat. Alright. You'd think that because of the name polymerase and the fact that they make nucleic acids, DNA polymerases and RNA polymerases would be related, but they're totally not. The genes that they come from have no similarity, well, almost all no similarity in their nucleotide sequence. So these two sets of enzymes likely arose twice during the evolution of early life, and one enzyme lineage led to the DNA polymerases, and as it turns out, reverse transcriptases. Reverse transcriptases look a lot like DNA polymerases. Another lineage of enzymes led to RNA polymerases, and they've diverged very heavily since, so they don't really look too much alike now. And when we look at our genome, a huge portion of our genome is transcribed, but not translated. So these primary transcripts, these pre-mRNAs, remember, are immediately destroyed and recycled, we think. And this explains actually some of the weird stuff that we see when we look at the human genome database on GenBank. So GenBank is an online database that allows us to look at DNA sequences, including the human genome sequence. There's a lot of these little primary transcript fragments left over. They're called EST tags. It's short for express sequence tags. So these RNAs we originally registered, we thought they were proteins, and so we thought they were genes. Remember, a gene is, for our purpose, anything that codes for a protein. But as it turns out, they don't really serve, as far as we know, any clear purpose. So we have a lot of these little leftover tags that we don't really know what they do. And as far as we know, they're just ghost RNAs that serve no function. Another interesting little bit is that RNA polymerases perform no proofreading. Therefore, we get large numbers of errors in transcription, and that is totally okay. Right? Because remember, we're going to be making hundreds, if not thousands, of RNA molecules per DNA copy. And if one of those RNA molecules is bad, no big deal. You get one bad protein, couple bad proteins, right? It's really when there's a problem in the DNA that it's a big deal. Now what this means, though, is that viruses that have an RNA-based genome that use transcription to make more copies of that RNA-based genome, they have a very high rate of mutation because there is no proofreading. So this includes viruses like HIV. HIV has one of the highest known rates of mutation out of any, I hesitate to say living thing because viruses are not technically living, but out of any living-like thing. There we go. It's all because of the lack of proofreading. Okay, so with these, right here, you see this? This is going to be extra credit on our next exam. You don't have to memorize every single one of these, but have a general idea, be able to recognize them. Alright, so I'll leave you to that if you would like. Finally, here's our summary. Look at prokaryotes. I know it's a weird spelling. I got this from another textbook. Just imagine there's a K there. So looking at prokaryotes, simple, easy, DNA to RNA to protein. Eukaryotes, on the other hand, man, there's a heck of a lot going on here. DNA, helps if I don't hit the end of the slide. DNA to our pre-mRNA or primary transcript. We have to go through capping, tailing, splicing, and export, and only then can we do translation. So eukaryotes are a heck of a lot more complicated. And that is it for transcription. Thank you as always for sticking with me. I appreciate it.